Welcome back to the deep dive. Really glad you could join us. Today, well, we're on a mission, really. We want to cut through the noise and focus on the really cutting-edge research, the clinical trials that are shaping the future of prostate cancer treatment. We've gone through a whole bunch of recent news, trial updates, all sorts of things. And the goal is simple. Pull out the most important nuggets, give you the insights into how science is fighting back against the toughest forms of this disease.
And it's such a crucial area. I mean, when you talk about advanced prostate cancer, especially the resistant kinds, it's a field that's moving incredibly fast. Our understanding of the underlying biology is just exploding, and that's really what's driving these innovations you're about to hear about. It shows a real commitment to finding answers where options used to be pretty limited.
Absolutely. So let's start with what's happening, well, right now or very soon. Clinical trials. This is where the rubber meets the road, right? Where new treatments get tested in actual people. First up, something pretty significant. The FDA, the U.S. Food and Drug Administration, has cleared the way for a first-in-human trial. It's for an NSD2 inhibitor called KTX-2001, and it's specifically for mCRPC, that's metastatic castration-resistant prostate cancer. A really tough one.
That's right. KTX-2001, it's an oral drug designed to be selective. And this is a phase 1 trial. They're calling it Strike 001. It's designed for patients, whose cancer has already progressed, even after getting at least one standard androgen receptor treatment. They expect enrollment to start sometime in the third quarter of 2025. And what's quite interesting is how they'll test it both on its own as a single agent and also combined with darolutamide.
Darolutamide, that's another established drug.
Exactly, another androgen receptor blocker. So the idea is hit the cancer from multiple angles at once, see if that combination is more powerful.
Okay, makes sense. Now, here's where it gets really interesting, I think. Moving to another aggressive type, neuroendocrine prostate cancer, NEPC. There's a phase 2 trial looking at a drug called Tarlatamab.
Tarlatamab, yes.
This is a fascinating immunotherapy. It belongs to a class called bispecific T-cell engagers.
Bispecific T-cell engager. Okay, break that down for us.
Think of it like a tiny molecular matchmaker or maybe a bridge. One end grabs onto a protein called delta-like ligand 3, DLL3. Now, this DLL3 is like a flag that's highly expressed almost exclusively on these NEPC tumor cells. The other end of the tarlatamab molecule grabs onto the patient's own T cells, the immune system soldiers.
Ah, okay, so it physically connects them.
Precisely. It brings the T cell right next to the cancer cell that has that DLL3 flag and essentially tells the T cell, attack this one. It activates the T cell to kill the cancer cell. And this trial, it's focused on adults who have metastatic NEPC, either from the start or treatment-emergent NEPC. These are often patients who've unfortunately gone through multiple other treatments already.
And the results so far?
Well, the early results look quite promising. There are side effects, things like low-grade cytokine release syndrome, which is common with these types of immunotherapies. But they seem to be generally manageable. And crucially, the preliminary data points towards a better objective response rate and also improved progression-free survival, especially in patients whose tumors are actually positive for that DLL3 flag.
So the target matters.
It really does. And it's also worth mentioning, tarlatamab recently got accelerated approval for a similar job in extensive-stage small cell lung cancer. That also highlights its potential in these neuroendocrine-type tumors.
That's really encouraging. Okay, let's shift gears a bit. A different class of drugs. CDK inhibitors, cyclin-dependent kinase inhibitors. We've got 2 new trials combining these with existing therapies for mCRPC.
Yes, and these are really aimed at tackling resistance again. First, there's a phase 1b2 study looking at TY302. This drug is a CDK4/6 inhibitor. Basically, CDK4 and 6 are enzymes that help control how fast cells divide. So this drug tries to put the brakes on cancer cell multiplication. And they're combining it with Abiraterone acetate.
Another standard therapy.
Right, one that targets androgen production. This combo is for mCRPC patients who've already failed other hormone-based therapies. The strategy is, again, a 2-pronged attack, disrupt the cell cycle, and cut off the hormone fuel.
And the second CDK trial?
It's similar but distinct. A phase 1b2 study of TYK-00540. This one's a novel oral small molecule inhibitor too, but it targets CDK2, 4, and 6. So CDK2, 4, 6, hitting a broader range of these enzymes simultaneously. It's being tested in combination with enzalutamide, another standard androgen receptor inhibitor.
Also for patients who've progressed on other therapies.
Yes, specifically mCRPC patients who previously failed novel endocrine therapies. The rationale here is that by targeting CDK2 as well, Maybe it can overcome some resistance mechanisms that pop up even with just CDK4/6 inhibitors. It's about, trying to outsmart the cancer's adaptability.
It really shows how complex fighting resistance is. Okay, and what about, well, another exciting area in immunotherapy, natural killer cells,
Yes.
or NK cells. We have an update on something called INKmune.
Yes, INKmune. It's an investigational immunotherapy, and it's had some really encouraging news from its phase 1/2 trial called CARE-PC for mCRPC. The trial actually hit its main goals, its primary and secondary endpoints. It showed a good safety profile and, importantly, signs that it was having a biological effect.
So what exactly is INKmune? How does it work?
It's quite clever, actually. It's basically a specially prepared pharmaceutical-grade human tumor cell line, but it's been made replication incompetent, meaning it can't grow or divide. Its job is to prime your resting natural killer cells, your NK cells. Think of it like sending your NK cells to boot camp. It transforms them into what are called memory-like NK cells.
Memory-like, meaning they're more effective.
Much more effective. They become better trained, more potent killers, specifically geared up to find and attack tumor cells.
And the findings from that trial?
Key findings were, first, it was well tolerated, no major safety red flags across the different doses tested. Second, and this is crucial, they saw NK cell activation, that priming effect, in over half the patients. Interestingly, this effect was strongest in patients who started out with low NK cell activity, which is useful, right? It helps pinpoint who might benefit most.
Yeah, that makes sense. Did they see any actual anti-tumor effects?
Well, the trial wasn't primarily designed to measure that, being an early phase study focused on safety and biology. But yes, some participants did actually see their tumor lesions shrink. And in a few cases, some lesions even disappeared completely. So that strongly suggests a direct anti-tumor activity.
Wow, that's pretty significant for a phase 1/2. What's next for INKmune?
The plan now is to move to a randomized phase 2b trial. This will likely be in patients with less severe disease, allowing them to really test its effectiveness more rigorously. It's a very positive step forward.
Definitely one to watch. Okay, so we've covered a lot happening in clinical trials. These are treatments getting closer to patients. But how do these ideas even start? Let's zoom out a bit. Look at the earlier stage research, the preclinical stuff that lays the foundation, and we have some truly fascinating discoveries here. Let's start with something totally different. A new bacterial therapy from researchers in Japan.
This one is really out-of-the-box thinking. It involves what they call the AUN Bacterial Consortium. It's a mix of two naturally occurring bacteria. Proteus myrtalis, they call that a Jew, and Rhodopseudomonas palistris, or UNGO. These are given intravenously, and here's the key. They seem to preferentially gather and grow inside tumors.
Bacteria inside tumors on purpose.
Exactly, and the truly revolutionary part. This therapy seemed to attack the tumor without relying on the patient's own immune system.
Without the immune system, why is that such a big deal?
Because many patients, perhaps due to chemotherapy or the cancer itself, have weakened immune systems. Standard immunotherapies that rely on boosting T cells might not work well for them. This bacterial approach could potentially be an option for those immunocompromised individuals. It's a completely different way in.
Okay, so how does it work then, if not through immunity? What did the animal studies show?
In animal models, once the bacteria establish themselves in the tumor, they cause pretty dramatic local effects, things like blood clots forming within the tumor's blood vessels, vascular collapse, the blood supply shutting down, and widespread death of the tumor cells. It seems to be driven by both starving the tumor of blood and oxygen, and also some direct toxic effects from the bacteria on the cancer cells. And importantly, they didn't see signs of this causing widespread problems in the body, like systemic toxicity or that cytokine release syndrome we mentioned earlier.
That's pretty wild. But using live bacteria, what about the control aspect? Isn't that risky?
That's a crucial point. What's interesting here is that these are naturally occurring bacteria, not genetically modified ones. And critically, their activity can apparently be stopped using standard antibiotics. That gives researchers a potential off switch, a vital safety feature.
An off switch, right.
But you're absolutely right to raise the question of risk. Using live bacteria therapeutically obviously brings up concerns about potential infections, maybe the bacteria developing antibiotic resistance, and long-term safety. These are all really important questions that need careful study before this could ever move into human trials. It's very early stage, but fascinating.
Definitely fascinating. Okay, let's circle back now to KTX-2001, that NSD2 inhibitor we talked about starting its phase 1 trial. What was it about NSD2 that made it such a compelling target in the first place? What did the preclinical work show?
Yeah, this really shows how that foundational lab work leads to clinical trials. NSD2 is an enzyme called a histone methyltransferase.
Okay, histone methyltransferase, what does that do?
Think of your DNA like a giant library of instructions. Histones are like the packaging proteins that help organize that library. Methyltransferases, like NSD2, act like little chemical tags, adding methyl groups to those histones. These tags help tell the cell which books in the library, which genes should be open and read, and which should be closed. Now, in healthy prostate tissue, there's not much NSD2 activity. But in advanced prostate cancer, especially the treatment-resistant types and those neuroendocrine variants, NSD2 levels are way up. It's overactive.
And what does that overactivity do for the cancer?
The preclinical research showed it does several bad things. It helps build the specific enhancer landscape, essentially setting up the control switches that allows the androgen receptor to keep driving tumor growth, even when hormone levels are low. It also seems to ramp up other cancer-promoting signaling pathways, and it can even contribute to making the tumor microenvironment suppress the immune system.
So it's helping the cancer survive and thrive in multiple ways.
Exactly. And connecting that to the bigger picture, when researchers in preclinical models like cell lines or animal studies silenced NSD2 or blocked its activity, they saw a sharp reduction in tumor growth and fewer metastases. So KTX-2001 is designed specifically to block that enzyme activity to stop NSD2 from adding those problematic methyl tags. The hope is that this collapses the faulty gene regulation program that sustains the tumor.
And combining it with darolutamide, the androgen blocker.
Right. The idea there is to hit two critical interconnected pillars of mCRPC biology at the same time. You block the androgen receptor signaling, and you block the epigenetic machinery, NSD2, that helps the androgen receptor keep working despite the blockade. It's a strategy aimed at preventing escape routes for the cancer.
It's really striking how many different angles scientists are attacking this from. When you look at all these approaches together, the targeted inhibitors like KTX-2001, the immunotherapies like Tarlatamab and INKmune, the cell cycle blockers, even the bacteria, what stands out most to you?
For me, it's really the sheer breadth and ingenuity of the innovation happening. We're seeing everything from therapies that target tiny molecular details within the cell to harnessing the patient's entire immune system in different ways, and even exploring completely novel biological agents like bacteria. But I think the common thread, especially for these advanced cancers, is the move towards combination therapies.
Right. Hitting it from multiple sides.
Exactly. Cancer is smart, it adapts, so combining different approaches is often necessary to try and overcome that resistance. And there's also a clear focus on finding better solutions for specific challenging groups like patients with NEPC or those who are immunocompromised. It's becoming more personalized, more tailored.
That's a great point. So for you listening, why should you care about all these, sometimes complex scientific details? What's the real relevance here?
Well, I think this knowledge is incredibly important because it shows the tangible progress being made, especially in areas like advanced prostate cancer, where options might have felt limited in the past. Seeing these completely new avenues being explored offers real, significant hope. It highlights how vital ongoing research is. It's this kind of work that ultimately leads to more effective, potentially more personalized treatments down the line. It means more tools for doctors, and crucially, more possibilities and hope for patients and their families.
Absolutely. It's about understanding the momentum, the direction things are heading. So in our deep dive today, we've really covered a remarkable spectrum, haven't we? From precision targeting inside cancer cells, harnessing the immune system, even using bacteria. The future for prostate cancer treatment definitely seems to be moving towards smarter, more targeted, often combination-based strategies, and that offers genuinely new hope.
It does. And maybe a final thought for you to ponder. Consider how all these incredibly diverse lines of research, targeting specific enzymes, priming NK cells, leveraging bacteria, they seem so different on the surface. Yet they all converge on one single goal. Trying to outsmart cancer's incredibly complex survival toolkit makes you wonder, doesn't it, what completely unexpected area of science might hold the key to the next breakthrough?
A really powerful question to end on. Thank you so much for joining us on this deep dive into the future of prostate cancer treatment. Stay strong and fight on.